Just waiting for everybody's audio to connect so we can get started. But first, let's start with thank you so much for being here. It's always great to see so many people joining us and being interested in what eButterfly is doing and how awesome scientists as Federico use this information that all of you guys are providing us to what we all are interested on, right? And preserving the butterflies all over North America. So most likely, most of you know me. My name is Rodrigo Solis. I am the Human Network and Data Coordinator for eButterfly. And I have the pleasure of introducing you to our guest speaker today, Federico. He joined as a postdoc fellow eButterfly about half a year ago, maybe?
Yeah, 6 months.
So he's been helping us and putting some science into our platform so we can orient whatever we do into conservation science. So anyway, without further ado, because he has a lot of things to say, I leave the floor to Federico. And yeah, take it on, Federico. Thanks.
Yeah, thanks, Rodrigo. Can you guys hear me? Okay, let me just share my presentation. You should see my presentation, correct?
Yep. Now we see it. Yes.
Okay, good. If you have any comment, please talk, because I'm gonna close my window and I'm not gonna see you, but I'm going. Okay, so yeah, thank you very much for showing up and for participating into this seminar. My name is Federico Riva. I'm a postdoc at Carleton University, and I'm working with the Montreal Insectarium and eButterfly for a MITAX Accelerated Postdoctoral Fellow. And today I'm going to talk about the importance of community science to support scientific decision in biodiversity conservation. I was born in Italy, where I did just a brief introduction about myself. I did my Bachelor of Science and my Master of Science in Italy, and then I came to Canada for my PhD at the University of Alberta. I'm a conservation scientist, and as part of my job, I do a variety of studies that range from landscape ecology to biodiversity science and biogeography, ecological theory, rare species. And one of the common points that connects all my research is the use of butterflies. So for me, working for a butterfly has been like a fantastic opportunity, and I'm enjoying every day of the process. And today, what I want to talk about is my presentation is focused around the importance of checklists, butterfly checklists that community scientists record and upload on eButterfly. And I divided my presentation in four sections. And at the end of my presentation, I hope to convince you that recording data as butterfly checklist will be important for us moving forward if we want to protect and manage biodiversity of butterflies across North America and across the globe. So the first part of my talk will be a brief introduction about what are checklists and the type of data that we can use in ecology. Then in the second part of my presentation, I will provide a couple of examples from my own work. In the third part of the presentation, I want to discuss briefly the importance of detectability in handling these checklist data sets. And at the end of my presentation, I will talk about some of my work with the butterfly and how I expect to incorporate my previous work in the analysis of the butterfly data. Okay, so the value of checklists. I am assuming that everybody knows what is a butterfly checklist, but it's still good to have a quick refresh. A checklist is simply a list of species that is compiled working a given distance in a given amount of time and recording all butterflies within a certain volume. So you can see on the top right, this scheme from the butterfly monitoring scheme in Europe. There is this imaginary volume around you. It's about 5 by 5 by 5 meters. And every butterfly individual of every species within this box is counted during the checklist work. The the important part of this type of data set is that one can record how long he has been working, how much time the checklist recording has been lasting for, if a person was by themselves or with somebody else, and the weather condition encountered while collecting the data. And all this information will be then helpful when analyzing the data to get the most out of the information collected. To take a step back and focus a bit more broadly on type of data in ecology, checklists are important because, differently from other data points like museum specimens that are associated just with the presence of one specimen of one species, allow scientists to make inference about species that were absent from a site or species that were non-detected, which are two different concepts that I will discuss later. So presence of data are great, and I also use them in some of the analysis that I will discuss later. But I hope that at the end of my presentation, we'll realize that checklists are better, and ideally we would like to start recording data with the highest amount of information possible. So why are checklists better? Well, to begin with, presence-only data are connected with just one species generally. So we have information about one butterfly found in one area of the planet in a certain date and the spatial coordinates. While checklist data include information about multiple species, and a given condition can use them to evaluate both presence and absences of species. For this reason, one finds easier to infer detection probability when using checklist data, or also estimating large-scale patterns of sampling effort, which is a very common issue with community science data. And last, it's very easy to transform checklist data into presence-only data, but you can never or very rarely transform a series of presence-only data set into a checklist. So at the end of the day, all these points are very important when we're doing scientific analysis of biodiversity data, because they allow us in having the most accurate inference about changes in space and time of the distribution of these animals. So after discussing what is a checklist, I would like to give you a couple of examples of how I've been using checklists in my personal research. I did a lot of checklists during my PhD and during my Master of Science. And I've been working anywhere from like the Italian Alps to the desert in Utah to the Alberta boreal forest. And on the right, you can also see myself going with Maxime Larivé here around Montreal. So I've been seeing a lot of butterflies and I have a lot of experience. And I think there are like aspects that I point out that are positive and negative. And I hope to discuss both of them today so that everybody has a better idea of what is the value of this sort of data. So I will talk about two quick study examples during my doctorate. And the interesting part about these studies is that the butterfly checklist can be already used as they are uploaded for this sort of analysis. So those are very important because they will allow major conservation gains moving forward. You will see later in this presentation that just in 2021, they've been uploaded 5,500 checklists in eButterfly. And in 5 years during my PhD, I collected 700 checklists. So the amount of information that is collected by community science is overwhelming and very useful. And so hopefully I can convince you to keep getting this sort of data because it will be very important to use. And a first very simple example of how I use checklists during my doctorate is simply in counting the diversity and number of butterflies in different types of forest disturbance, and use this information to suggest the best mitigation practices for the extraction of oil sands in Alberta. So the question is very simple. There are different types of forest change in this forest. You see some of these corridors are narrower, some are larger. There are some well pads, which are basically clear-cut patches. There are roads. And we didn't really know how insects respond to this forest change in Alberta. And we use butterfly as an indicator of change in this forest. And simply by collecting checklists into these different types of forest disturbance, control forest, narrow seismic line, narrow cut lines, and larger cut lines, we were able to detect at which point anthropogenic changes in habitat are actually triggering a response in the forest environment. So you see that between a control forest and a 3-meter wide cut line, there is virtually no difference in the diversity of butterflies. We found about 7 butterfly species in these checklists. But as soon as we moved to a 10-meter wide corridor, like the one on the right side, we found almost twice as many butterflies. So this is a very simple example of how just butterfly checklists can inform management. And community science data could have been used to answer this question, but unfortunately they were not available. Another example of my work using checklists, also in the boreal forests of Alberta, is a study that investigated the relationship between plants and butterflies in a system where there are overlapping disturbance types. So in 2016, there was a very extensive wildfire in Alberta near Fort McMurray that burned 6,000 square kilometers of boreal forests. You can see it in dark brown here. And these wildfire provided the opportunity to study how unnatural disturbance regimes like fire interacted with these very widespread forest disturbances. You can see all these linear cut lines across the forest. Some were burned and some didn't. And so we wanted to understand how the overlap of wildfire and anthropogenic cut lines was affecting butterflies in the boreal forest. The first most staggering result that we noticed is that even though the forest around cut lines were completely burned, like 90% of the trees basically burned to death, we found almost the entire cut line soil surface unburned. And this suggested that basically fire was not burning severely enough to damage the plant community at the ground level in these disturbances. And unsurprisingly, we found that there was a positive correlation between the diversity of plants and the diversity of butterflies in these sites. And we found that burned seismic line, you can see in this orange pink, virtually had the same diversity, if not slightly higher, than unburned cut lines, suggesting that these anthropogenic corridors did not really burn as the adjacent forest sites that we studied. As I said before, checklists are important because they provide information about a multitude of species. And this is just an example of the type of analysis that we can do. At the bottom in purple, you can see 30 butterfly species that we found in these boreal forests and their response to cut lines and wildfire. And yeah, the take-home message of this study was that even though anthropogenic changes in boreal forests are not necessarily natural, we found that in the face of severe wildfires that are expected with climate change, there is still a refuge effect, in a sense that butterflies were able to persist in these environments. And those are just two very simple examples from my research, but there are like very many studies that use checklists across the globe. There is a very cool study by Tasman Webridge in Ohio that showed biodiversity declines in the number of butterflies counted in the past 20 years. There are studies on trends in phenology, which are anticipating. And in Europe, it's even integrated in like more formal policy frameworks. So there is like a grassland report that is compiled periodically by Butterfly Conservation Europe. And here on the right side, you can see a distribution of monitoring sites that are visited multiple times. And this sort of information is vital for conservation. And my hope is that with the Butterfly, we'll be able to establish this sort of effort that is continuous in time and provide us with information that we can analyze to understand trends in space and also in time. Okay, so we talk about what is a checklist and we talk about how we can use them. But now I would like to discuss a bit some of the lessons that I learned by using checklists when I started looking at the assumption behind this approach. I'm assuming that everybody in this talk has been walking around and looking for butterflies because we're all part of this big family. And I know that everybody goes to a site every once in a while and expects to see a butterfly and we don't see them. And this can be surprising at times, and we don't really know if the butterfly went extinct in that site, if we were in the right day or if we were in the wrong day, if the environmental conditions are not ideal, and so on. And analyzing checklist data without taking into account the problem of imperfect detection really assumes that everything that was there was observed and everything that is missing was not present in the site. However, we know that we're not 100% sure of seeing all the butterflies in a site when you're working there. And there are many examples of cases that are crystal clear. For instance, if you're working in December in a grassland, you're not going to see butterflies simply because at that point of the year, they're not easy to recognize because they're not flying. And during the day, if you're working at 4 a.m. in a meadow, you're not going to see butterflies because that's not the moment in which they're active. Different observers can have different experiences. Some species are easier to detect, like a swallowtail is going to be much easier to detect than the species here on the right side of this slide. Habitat type has an effect, and so on. So I did spend some time during my postdoc working on these sort of issues. And I'm going to talk today about 2 studies quickly that highlight the problem that we have when we assume perfect detectability. The first one takes a common assumption that if I want to understand which species inhabit a site, I have to visit every 10 days or a week. With 6 or 8 visits in 1 year, I should be able to have all the information I need about the biodiversity of that site. The second study evaluated the assumption that in recommended sampling conditions, so between 9 a.m. and 3 p.m., when temperature is warmer than 17 degrees and when wind speed is low, the number of butterflies that I see in a site is not affected by the environment, but just by the quality of the site itself. So how often should we sample butterflies? This was a question that I really took at heart because in my doctorate, I went for one study 8 times during the summer and for another study 12 times, which is approximately once every 2 weeks or once a week, which is on the intensive side of sampling for this sort of studies. But for this specific study that I did with Leonardo Vigliani, which is a student in Italy, a master's student, we selected 5 sites and we sampled them once a day, every day for 3 weeks. So this was a very intensive sampling design, which provided us with 25 observations per site within recommended sampling condition. Then we selected a random number of, randomly a fixed number of these pollard walks, this checklist. And then we evaluated how well they represented the sites based on the total diversity of butterflies that we notice in this site as a reference. I'm just going to present results for species richness for simplicity. But imagine that on the y-axis, you have the proportion of missing species in comparison to the true state of a site. So the closer we get to 0, the better is our understanding of the diversity of that site. And on the x-axis, there are like the 8 different type of simulations we run. 1 transit every 20 days, 1 transit every 10 days, 1 transit every 7 days, 1 transit every 5 days, and then 2 transit at the same frequency. What we notice is that with 1 visit every 10 days, so 2 transits in 20 days, on average, we detected just 50% of the species present at the site. And this was surprising because most of the studies that use checklists tend to recommend 1 visit every 2 weeks. And sometimes they go up to 1 visit a month. And even when we had 8 visits in 20 days, which is way beyond any experimental design that I've seen in the literature, we still miss 20% of the butterfly species. Here we have both bees and butterflies, but you can see that butterflies are in gray and the 2 groups are very similar in their response. So again, on average, we miss half of the species with a visit every 10 days and 20% of the species with 8 visits in 20 days. So these outline a problem that is the problem of imperfect detection. That even within a day, even though we know that there are going to be butterflies inhabiting a certain site, there are other factors that are affecting our ability to see them. For instance, if it's a windy day and it's cloudy and it's cold, we know that butterflies are going to be less active than in a sunny day. And that affects the number of butterflies that we can count. So I did a second study where we specifically evaluated how the environmental conditions within one day are affecting the number of butterflies that we can count. And this is a result that Maxime told me to discuss in detail, because I know that we are all looking for the best moment to go out and catch butterflies. So I hope you will be interested in this. But imagine that we have 10 butterflies, and on the y-axis of these graphs, we have the probability of seeing them. So if we go out in the field for this specific species that I studied, which is called Arctic fritillary, if we go out in the field in a normal day that is sunny and has no wind, at midday, the probability of detection will be approximately 40%, and it's completely independent from temperature. During sunny days, the most important factor explaining how many butterflies I count was the hour of the day. So the probability increased between 9 a.m. and midday, and then eventually declined later in the afternoon. This, however, wasn't the same for cloudy days. So during cloudy days, temperature had a much stronger effect. And what is interesting is that it is recommended to start sampling at 17 degrees when it's a cloudy day, and the temperature is just enough for butterflies to fly. And we found a change in the probability of detecting butterflies from 10% to almost 50%. So to give you an idea, if I counted 10 butterflies with 17 degrees in these specific conditions, they were probably 50. And this is an important factor that it's generally underestimated in the study of checklists, and we hope to integrate more and more in the study of butterfly. And we can do this only with very extensive data sets. So this is just like a funny slide. And on average, detectability in my study was 20%. So for every butterfly that I was catching, there were like 4 of them behind me just looking at me. Okay, so now we are talking about the project that I'm working on in eButterfly, which is very exciting. And again, I'm assuming that everybody is familiar with eButterfly, but to me as a scientist, having the possibility of working with eButterfly is really amazing and exciting, because it's an incredible resource of data points. Just in this past year, there were like 5,500 checklists uploaded. There are more than 100,000 checklists in the past 20 years. Thousands of observers that I want to take the chance of thanking again. And I do believe there is like a strong potential to leverage this data to recommend conservation policies and management of biodiversity in North America. But there is also some work to do, and here is where I hope to be able to contribute in the next few months. And the first project that I've been working on is creating range maps for all the butterflies of North America. As I was mentioning, eButterfly allows you uploading data sets as a checklist. So a series of species found at a site and in a specific time of the day. And this is an example of a checklist where you're gonna have like 42 monarchs and 6 cranberry blue butterflies and so on. But as I said before, the importance of checklists is that we can somehow infer absences if we have the right data to do so. And one approach to produce the absences based on checklists is to create a species pool for basically every site in North America. And then we can use the species pool, which are the species that to some extent we expect to occur there, and use that to infer which species were not seen. So maybe in this case, we counted 1, 2, 3, 4, 5, 6, 7 butterflies, but we know that actually in the site during the year, we can have maybe 45 species. And so we can create a list of 38 species that were not observed and use that in our analysis. But these require creating range maps for all the butterflies of North America. And that has been my first task here with the butterfly. And I will not go into the technical details, but the process is very simple in that we have occurrence data. So you can see the points here for the disalpine, which is kind of a northern species that I selected because it has a very strong effect of temperature on the distribution. It tends to occur just in very cold areas. And we combine this data on the distribution of the species with data on climate and data on land cover. Here I just have 2 examples, but we have much more information. This is like the amount of degree days in 1 year multiplied by 100 and the proportion of forest. So you can see, for instance, California and Mexico have more degree days than Alberta or like Canada, and the proportion of forests is just it's a remote sensing product. So there are like satellite pictures that allow inferring how much of the continent is covered by forest. And by combining this information, we can create these surfaces that somehow represent how suitable is every one of the 660,000 5-kilometer cells that create North America. And this suitability map with some statistical techniques can be converted into a binary map. So for example, for this specific species, the species is predicted to occur in this green area here. What is interesting is that we do have climate data for the present condition, also for the future conditions, and also land use data. So you can see this is a map based on the data available between 2000 and 2020. And we can extrapolate the relationship between the species and climate to 2050. And you can see that there is a contraction in the distribution and a shift toward the North Pole. So we do have this information for about 640 species, which is amazing. And it is going to be very useful because we know which species of trees and birds, for instance, are going to be more exposed to climate change. But we don't really know across North America where we should protect, where we should create new protected area or like recommend additional mitigation measures in areas that are the most exposed to climate change with the highest biodiversity. And so we are working to create this sort of maps that will inform conservation planning in the next 20 to 30 years. And just to give you an idea of the amount of data that we have available, by combining eButterfly and GBIF, there are more than 360,000 observations for the 740 species of butterflies available in North America. I just want to take very quickly the chance to highlight a few species for which we're missing data. So those are 90 species for which we have less than 10 observations. You can see that most of these species have plenty of data, but these species are species for which information is scarce. I don't want to spend more than 10 seconds on this slide, but we will upload it online on eButterfly on the website, and we will create like a contest, and we'll ask users if they know of sampling sites that actually have these species, so that we can have the best possible definition of distribution of these species as well. And after doing this exercise of predicting distribution of species, we are going to have information on which species were present and absent for all the checklists that we created. And so I want to work on population trends. There is this very interesting study by Tyson Weprich in Ohio, and they showed that there were declines in the past like 20 years that resulted in almost a third loss in abundance. And my idea is to sort of replicate these studies for Canada, potentially accounting for detection probability, as I said before, because that's something that has not been done, but I think might show some interesting pattern. And another study we are thinking about doing is replicating my detectability study in the insectarium greenhouse experiment. So every time you're in the forest, you don't really know what is the true number of butterflies, but in a closed environment, you are going to be able to manipulate the number of butterflies, maybe the temperature, some other factors, so that we can get the best possible information on what really makes easier to see butterflies in the field, also to recommend the sampling condition for different species. So in conclusion, why does this matter? Community science is perhaps one of the biggest hope that we have in conservation in our challenge of facing the current biodiversity crisis. You know, we are living in a time of change. Temperature has been increasing basically in the past 150 years. Habitat loss has been it's increasing globally and it's widespread. And there is no way we can protect biodiversity unless we know where species are and how they're changing. And butterflies have been used for decades as an indicator of environmental change. And they're considered kind of a canary in the coal mine, in the sense that having these very short life cycles, they can respond quickly and at a very small scale. So checklists from community science will be crucial for biodiversity conservation, and they have already been used extensively in Europe. So my hope is that eButterfly will kickstart this approach also in North America. And I want to close with like a very personal insight. as a butterfly scientist, I do feel like we have a responsibility because butterfly has such a strong leverage on common people. And this is just a picture I took from like a butterfly day when I was in Edmonton. And we had probably 100 kids just dressing up like butterflies and, starting ending their nets. And this is a picture I did when I wasn't even able to write. I think I was like 3 or 4 years old. And yes, there is a car, there is a house, but then there are like plenty of butterflies everywhere. And I do think we have the responsibility and the opportunity of leveraging this interest that people have for these magical animals that myself and a lot of you appreciate and love, and hopefully we'll be able to protect them. And yeah, that was the end of my presentation. I just want to acknowledge my funding sources, especially MyTax and the Espèces pour la Vie, the Museum Montreal and Carleton University. And at the bottom, there are some other sources that funded me during my doctorate, so the studies that I discussed before. I want to thank Maxime Larivé and Lenore Ferrig, which are my co-PI that are supporting me during this project. And also I take the chance of thanking Scott Nielsen and John Echo, who supervised me at the University of Alberta, Jaime Pinzon, who was a co-author in the plants and butterfly study on the Alberta burn cut lines, and Leonardo Vigliani, my student in Italy, who did all the 150 transects in 20 days, which was amazing. And with that, I thank you, and I will take any question.
So thank you so much, Federico. This was great, and I think it's always quite empowering to know, just as a eButterfly user, to see how far our observations can reach, right? So it's not only the enjoyment of going out and registering butterflies, but actually how it can save them, per se. So thanks a lot, Federico. And just one last thing, remember that we have different types of submission on eButterfly. We have time count, we have distance travel, we have area count, and each one of them have a pros and cons. So I don't know if Federico, you want to
yeah, so I talk about checklists for commodity, but yeah, of course, if we have like a point count, we can treat it as a checklist. So as long as we have information about multiple observation counted and the time that the sampling was lasting for, it's equally important. The main difference is between just recording one specimen of one species in space in contrast to a list of species that was sampled in any different ways that can be recorded. So point counts are also equally valuable. Yeah, I try to simplify the presentation as much as possible, but these different types of samples, as long as they have like a measure to quantify effort, can be integrated. Even different types of sampling can be integrated in the same model, so they're fantastic.
Okay, perfect. Thanks a lot, Federico. So we will open the Q&A session now. So for sakes of security, I will unmute you. So please, if you have any questions, just raise your hand. You can do that on your lower right part of your Zoom screen. There's reactions. So just click reactions and make raise your hand or whatever, and I will unmute you so you can do your questions, or you can also paste them on the chat, and we'll be happy to ask them to Dr. Federico.
Federico, I see there's a couple questions in the chat. I'm going to read one for you here. Paul wrote, could you address the merits of entering a time count as opposed to a distance travel count? I know we just sort of touched base on that a little bit, but there is a difference there. And from your perspective, what is that? Is it better to do travel if you can, or what's the good, the bad, and the ugly of that?
Yeah, so ideally, if we have information on sampling effort, one can integrate them in a statistical way. So they're both equally useful. But traditionally, checklists are, polar walks have been designed to cross different environments. So by walking around, you can see more species and you can get like a better information. But there is really nothing that makes one method better than the other, as long as you have information on multiple species and on the time spent collecting data, the environmental conditions. So that would be my answer. And my bad, I probably have been superficial in saying checklist, but what I was referring to were mostly list of species collected, accounting for how long the sampling was lasting for, and the environmental conditions and so on. So point count are also useful and can be integrated in this type of analysis that account for detectability. I don't know if I answered the question or.
Yeah, one thing I'd add to that too is that what strikes me about distance travel counts is that we just have the point that the user gave us. Versus with distance traveled, at least we know, like if you put down, oh, I traveled, 1 kilometer, we know you went a pretty good distance while you're doing that, and so we have a little bit of spatial error around, or spatial knowledge around how big of an area you're in. If we want to do something spatially with it. So I guess that's one of the other things I was thinking about with that.
Yeah. So all the analyses that I'm running are at the resolution of 5 kilometers, which is generally bigger than the distance traveled by the observers. So that was a way to account for that. But yeah, of course, if you have a point observation, you can get a higher resolution, like you can get model at a higher resolution, for instance. And again, I think the main limitation right now to make this sort of model is the amount of data. So ideally, we would like to integrate all these different sources. And yeah, I mean, again, the more data we can get, the better it is, and point count are equally valuable.
Perfect. Thanks, Federico. We have another one, question from Emma Hutchins. Thanks a lot, Emma. And she says, thanks for the great talk, Federico. Do you see any synergies between your recent advances and those happening with other community science tools like eBird, especially with regards to detectability?
Yeah, thanks, Emma. Yeah, eBird has the fortune of having an amount of data coming in that is unmeasurable. Like the amount of people submitting their data makes it much easier for them to have a sample size sufficient to run these analyses. But the principle is the same. So creating these binary maps of distributional butterflies was inspired by what they do to account for detectability. So we can feed these dynamic occupancy models, which allow accounting for detection probability when estimating the probability of occurrence. So they are at the forefront. They have a very strong analytical team and with professors working on them. So they are kind of opening the path for other community science platforms, and I'm following most of the work they've been doing.
Perfect. Thanks, Federico. Then we have a question from Tyson Webridge. Hopefully I said your last name correctly. Great to hear about your work. In addition to range maps to inform absences, do you have plans to use phenological curves to know if absences are seasonal?
Yeah, so that was one of the reasons why I wanted to create these binary maps, because we can start using time as a covariate in the detection probability. So if you're modeling the probability of seeing a species and we have week of the year, we might know that a species occur in Alberta, but maybe just occurs in July and a sample was made in June. And so we can account for that by just having a part of the model that is a function of time. And so my way to tackle phenology would be incorporating it in the detection part of the model, which is what we did at a much smaller scale with the Arctic Fritillary model, because we had a change in abundance that was a function of day of the month. And that's how we estimated the effect of temperature and wind and presence of clouds. So yeah, basically we can expand the model that already fit before to have a parameter that affect either fits a unimodal or a bimodal or a trimodal distribution to account for the phenology of this species.
Perfect.
So just to follow up on that, Federico, so you can have different modal distributions based on how many flights there are per year, to fit the model. Yeah.
So the simplest way would be for species with one flight curve, because you just have two parameters like a linear effect and a quadratic effect, which makes a normal distribution. And you can go in different ways. You can either subset the season, like you can have like 3 models, one for June, one for July, and one from August, and then assuming that you will have sort of normal distribution, or depending on the phenology of a species, you can kind of clip parts of the fish season. So there are different approaches to do that. But yeah, phenology is definitely something important that we are going to incorporate in the models.
Perfect. Thanks for that, Federico. Now we have one question from Paul Prapes. He says, allowing for errors in identification allowing for errors in IDs, are entries without photos still used and valuable? Would you rather someone enter at the genus level if the species is not 100%?
Okay, the photo would always be great. We didn't incorporate any observation in these models for which we didn't have the species ID. So we filter data, all the data for which we didn't have information on the species level, and also for which accuracy wasn't at least, I think, 4 kilometers, which is slightly smaller than our raster cell. So we really look at the quality of the data. And that's why we had 90 species for which we don't have really too much information. Because to begin with, in North America, we do have very good information about the distribution of these butterflies. But when you want to look specifically at a very fine scale, so a very high resolution, like a 5 by 5 kilometer cell, then you need to have data that are at high spatial resolution and also high quality for the taxonomy. So I don't know. I mean, I think there will be application also for data points just at the genus level, but for the projects I'm working on right now, those data points will not be used. So ideally, if you can get a picture, that's great. Yeah.
So just giving a quick parenthesis here, just remember that our database has tons of identifications. Some of them are already identified. Some of them are still to be identified. And our processes of identification is crowdsourced. So actually, you guys are the ones that help us going in, checking those photos and identify them and to vet that that species is the ones that the observer submitted. So yeah, like hopefully over time that database will just keep growing because of you guys identifying. So just quick plug in there. Max, you were raising your hand.
Yes, can you hear me?
Yep.
Okay, so I also wanted to add to this that we haven't done it yet, but we are also planning to be able to score each occurrence that is non-photo based on the amount of images, the amount of photos vetted by the user, by likelihood of other occurrences of the same species being present and have been submitted from a spatial temporal standpoint as well. So those, so ideally, we want a species ID when the ID can be done by a user. When he's uncertain, obviously, we'd rather have a genus ID or a family ID, but when they're confident about their ID, even though it's a sighting, because we can start adding weight to the likelihood that the identification is good based on all kinds of other covariates. Anytime you can go on and submit a species ID for your sighting-based observations.
Exactly. And on top of that, Max, one thing that maybe most of you already have heard a little bit about it is that we are pretty close to launch an image recognition algorithm that will help us to automatically identify and suggest that those tricky species. So that's going to help us to grow our vetted database. Anyway, I don't have any other questions here on the chat. I don't know if any other person has any questions that you can raise your hand anytime, or Federico, if you have any final words.
Yeah, no, I want to thank all these people that showed up. It's a pleasure. I see some like names that I recognize. Thank you for coming. And I just want to add that for the AI, for this algorithm that will help in classifying species, these maps that we are creating, actually, we provide some special filter to help in identifying species. So we're going to have like a special filter, and also we're going to have like a temporal filter. So we are working to incorporate all the information that you are providing us to make it, you know, as good as we possibly could and as fast as we can.
Exactly. Okay, perfect. So if we don't have any other comments, questions, please always, if you come up with any other questions, feel free to maybe shoot me an email to ebutterfly.help@gmail.com. I don't know if Federico, maybe you have your